This lecture will introduce you to some basic principles of pharmacodynamics. What I'll do is show the pillars of PK/PD and QSP, provide basic terminology and definitions, make comparisons of PK versus PD, show you nonlinear receptor target binding concepts, turnover concepts, and indicate the array of PK/PD models that can be encountered. So the pillars of pharmacokinetics and dynamics start with pharmacokinetics itself, pharmacology, and physiology. Underpinning these pillars are the foundation, the fundamental laws of biology, physiology, and pharmacology, some of which I indicated previously. And then we need tools in this edifice: mathematics, statistics, computation. All of this comprises pharmacodynamics, PK/PD, the science. So an edifice is an impressive structure, a concept system of beliefs. And that's what this field basically is. So pharmacodynamics is defined as a description of the time course impacting controlling drug effects on the body. We seek to identify capacity constants, sensitivity constants, and a whole variety of time constants that pertain to specific models, particularly related to turnover. What you see on the right are some PK/PD data. Plasma concentrations of erythropoietin after various subcutaneous doses, and time course of reticulocytes that are stimulated by this compound. So what happens is that the drug, erythropoietin, also a natural compound, acts in the bone marrow to stimulate the progenitor cells. There's a cascade of things that happen in the hemopoietic system. Ultimately, the reticulocytes appear in the blood and get converted into red blood cells. So we see that we need the bone marrow and blood compartments as aspects of pharmacodynamics. And we see that there are processes in between all of these. But like in pharmacokinetics, we can convert the anatomy and physiology into compartment-like models. So the bone marrow is essentially a precursor compartment, the blood is a response compartment, and we have the transfers and conversions and elimination steps that take place as well. So what's good is that we can represent pharmacodynamics as compartment-like models that allow better understanding of the processes taking place. So let's compare PK versus PD. Pharmacokinetics, there's a whole body influence. You give the drug, and most distribute throughout the whole body. Pharmacodynamics, we're often looking at specific subsystems, like I just showed. Pharmacokinetics, most of the time there's a zero baseline. There's no drug until we give it. Pharmacodynamics starts from steady state, some equilibrium situation. Pharmacokinetics has what might be considered simple determinants. The controls get far more complex than pharmacodynamics. Pharmacokinetics is usually but not always linear. Pharmacodynamics is predominantly nonlinear. In kinetics, there's few assay methods and media. We assay blood and tissues and excreted materials. In pharmacodynamics, there's diverse measures, all sorts of biomarkers and physiological responses. However, comprising both PK and PD are components and processes that can strive with models and equations, allowing conceptualization, quantification, and prediction. So besides pharmacokinetics, basic tenets of PD are the idea of capacity limitation and turnover and homeostasis. So the law of mass action, drug binding to targets, and the small quantity of targets leads to capacity limitations in most responses. Basically, drug content can be unlimited, target content is very small. So we see that this produces capacity limitation, and we see relationships like the Hill function that reflects this law of mass action. For turnover and homeostasis, we're dealing with biological factors or structures or activities where there's production processes and loss processes. The production processes, most simply, are zero order. The loss processes, most simply, are first order. So that presents a simple paradigm for turnover and represents a basic starting point for many of our pharmacodynamic models. In terms of targets, the kinds of receptors that we deal with are shown on the slide. So we have receptors that permeate cell membranes, such as G-coupled protein receptors. Some of our drug targets are enzymes. Some of them are nuclear receptors. Some are ion channels and DNA itself. So all of these biological entities are present at low concentrations and often can be saturated. The idea of nonlinear drug receptor binding originated with Ehrlich and Clark. Ehrlich coined the term receptors, and using Latin, he said, corpora non agunt nisi fixita. Substances do not act unless bound. The kinds of binding relationships that you see are reflected by the law of mass action. A ligand L binding to a receptor, producing a complex, and then producing an effect. So the drug receptor complex can be represented by rate constants for binding and dissociation, K1, K-1, or equilibrium constants like KD. So a nonlinear equation for drug receptor binding might have a Bmax and KD value in there, a capacity constant and a sensitivity constant. If we're looking at the effects produced by such target binding, we might then encounter the Hill equation or the Emax equation, where the capacity constant is the Emax and sensitivity constant is EC50. Both these equations are nonlinear. They're nonlinear because of the presence of the drug concentration as a variable in different situations in the numerator and denominator. So as drug concentration changes, there is a nonlinear behavior of the resultant response. We can see that when we look at receptor binding. These are data from my lab where we examined prednisolone binding to glucocorticoid receptors in rat liver. So the paradigm for corticosteroids is that they distribute from plasma into cells. In the cytosol, they bind to receptors. The receptors get activated, translocated into the nucleus. There, they alter the production of various messenger RNAs and proteins that comprise the whole array of kinds of effects produced by corticosteroids. But fundamental is nonlinear binding of the drug to the receptors. And what you see in the graph is the result of our isolating the receptors and adding a radioactive drug and being able to measure receptor-bound drug versus free drug in the system. And you see that it's nonlinear. As the drug concentration increases, ultimately there's a plateau. So the plateau is Bmax. On the graph, you can see that it's 2.5 nanomolar. Km can be identified as the concentration at 1/2 of Bmax. And interpolating the graph, it's about 80 nanomolar. So many drugs bind in this fashion to receptors or targets, where there's nonlinearity in binding and a basic equation with a Bmax and Kd value can be applied. The other aspect of pharmacology is looking at the overt effects of the drug. When we look at some endpoint in drug action, the Hill equation is often applied. The Hill equation originated from A.V. Hill when he was studying binding of oxygen to hemoglobin. And you see at the top, the Hill function has an Emax and EC50 value, a capacity and sensitivity factor, in the same position as Bmax and Kd. But there's also a power coefficient, gamma. The gamma term is added to the concentration term. So Hill needed this gamma term because of what's called cooperativity in the binding of oxygen to hemoglobin. It basically says that the speed of binding changes as the drug concentration changes. So it produces curvature, extra curvature to the curves. So here when we look at the graph of effect versus concentration, in essence, all of these curves should go up to 100. They ultimately will at high concentration. All of these curves go through the same point, the EC50, which you look on the graph, you can see that it's about 15 of these concentration units. But the different values of gamma cause a twisting of the curves, so that some of them are more shallow, some of them are more steep, depending on the value of gamma. So sometimes we need gamma in describing the nonlinear actions of drugs. A simple example of operation of the Emax equation is in this slide. So these investigators from Buffalo gave metoprolol to male and female subjects. Metoprolol is a 2D6 substrate, but also as it happened, there was a difference in clearance of metoprolol between men and women. So you see the plasma concentration versus time curves in the top graph left, and you see the calculated clearance on the bottom. The men data were lower concentrations than the women. The clearances were twice as high. But metoprolol is a beta blocker. It reduces heart rate and blood pressure. So the graph on the right is showing reduction in systolic blood pressure in the males and females. So this is not time course of data, but it's where they related the effect to the plasma concentration. So you see the curves, and you see at the high concentration, the Emax in both men and women is about 18, and we can interpolate at 1/2 of Emax, the EC50 value is 28, 25, about the same in men and women. So this shows the simple version of the Hill function operation of Emax and EC50 for the cardiovascular effects of this beta blocker. We will be describing many different types of capacity-limited relationships in pharmacokinetics and pharmacodynamics. Many of you have heard of the Michaelis-Menten equation for nonlinear drug metabolism. We'll be using that in pharmacokinetics. Shannon described a similar equation for transport. We will be looking at plasma protein binding concepts and apply the equation from Goldstein, the third equation that you see. I just showed you receptor binding and pharmacological effect equations that comprise parts of pharmacodynamics. And lastly, there's an equation for transduction that will be introduced later in pharmacodynamics as well. All of these equations have the same general structure. What you see is a capacity constant on top, and in the denominator, there's some type of sensitivity constant. They're in the same position in all of these equations. So when you learn one system, we'll be starting with protein binding after this lecture. You'll find relevance to other aspects of this form of nonlinear equation. The third pillar of PK/PD modeling is transduction. I'm sorry, the third pillar is turnover. So when we consider turnover, we can look at various body processes and see different rates of turnover. So some of the fastest turnover rates are electrical signals and EEGs. So one knows you can see an EKG as pictured, and the technician can identify heart rate and the nature of cardiac functioning very quickly, because the turnover rate is milliseconds. The same thing happens with neurotransmitters in the synapse of some cells. When we look at chemical signals, mediators, electrolytes, the turnover gets to be in the order of minutes. When we deal with a hormone like cortisol, the turnover is on the order of hours. Cortisol has a half-life of 1 hour. When we look at gene expression, messenger RNA turnover is often minutes to hours. Proteins and enzymes are similar, hour time frames. Cells have time frames of hours to days to weeks. Tissues, time frame of a month. Organs, a year. And people turnover. Most of you will have an expected lifespan of 1 full century. If we compare a couple of organs and tissues, we can consider muscle and bone. Not everybody realizes that muscle tissue is very active metabolically. It actually turns over every 2 weeks. Every 2 weeks, your body has degraded components of your muscle and synthesized new materials. Bone is much, much slower. Bone turns over every 10 years. It's a very, very slow metabolic process. We need to keep these turnover rates in mind when we're considering the actions of drugs and doing clinical studies. The early part of the list typically are biomarkers that we use to monitor actions of drugs. The bottom part of the list tends to be the clinical symptoms that can be derived when there's alterations in disease processes. Also, we use turnover rates to identify our models. When turnover is fast, we will apply direct effect models. And what's happening there is that the drug kinetics mostly control the pharmacodynamics. That's because the turnover is fast, so as drug concentration changes, the organ function, the EEG signal, will change in accordance. The middle part tends to be what we call turnover models. It's where the production and loss process in addition to the pharmacokinetics, will control the action of the drug. And there, towards the bottom, we need to invoke transduction models, where there's a constellation of processes that describe actions of drugs. Overall, if we try to incorporate a multiplicity of factors controlling drug action, we might evolve systems models. And systems models, as I previously introduced, try to incorporate a wide constellation of information into understanding the nature of drug action. An example of the need to consider turnover is shown here. These are data from a clinical trial done with Avandia. So what's plotted is glucose concentrations in diabetic patients. So the top line basically is showing over a period of 26 weeks, half of a year, that the patients being treated with metformin had a slight rise in glucose concentrations. But the patients treated with Avandia, a lower dose and a higher dose, gradually attained much lower concentrations, a lowering of 53 concentration units. So this is indicative of turnover being modest, somewhat slow. The turnover represents the turnover of GLUT transporters in muscle and other tissues. The drug is taking time to increase the tissue content of GLUT transporters, thereby resulting in better tissue uptake of glucose, thereby lowering blood glucose concentrations. So appreciating the turnover rate of GLUT transporters is essential in designing clinical trials of drugs such as this. In dealing with PK/PD, we consider Gerhard Levy, our faculty member, Gerhard Levy, as the father of pharmacodynamics. He made many different contributions to pharmacodynamics, as you will be told in this course. But he first identified what's known as the Levy-Km equation. So this was the first equation that accounted for the time course of drug action. So what you see on the graph is simple pharmacokinetics, natural log of concentration versus time. There's a decline in concentrations with a rate constant K, or KEL. When we look at pharmacology, we can look at effect versus the log concentration, and we see that in the middle of the curve, in the middle of the hill function, in fact, that the data will be almost, the line will be almost linear, and he called that the M slope. And then he noticed that when he looked at the loss of pharmacologic effect, a linear scale for effect versus time, that the slope of the graph was zero order. The zero order slope was minus K times M. The slope is the constellation of a contribution of pharmacokinetics, K, and pharmacology, M. So that defines loss of drug effect as a constant rate, zero order. This will, in fact, apply to most of the models that we will encounter in pharmacodynamics. The kind of data Levy analyzed to display these concepts is pictured here. These are data for succinylcholine. They gave an IV dose of the drug, and what you see in the graph is degree of muscle relaxation. So one can see a plateau in the graph, the Emax of 100% muscle relaxation. And then you see the decline phase. That's the Levy-Km slope. So you can see the data are plotted on a linear scale for effect, a linear scale for time, and you see the nearly straight line relationship that Levy called Km. Well, it's not perfectly linear. There's slight curvature to it, but it approximates as a linear decline over time. Wagner then contributed further to pharmacodynamics. He took what Levy showed, and what he did was to identify the fact that the Hill function was useful in pharmacodynamics. The Hill function was previously used for oxygen binding to hemoglobin, but Wagner showed that it's a relationship that generally applies in pharmacodynamics. And then he used simulations to indicate what might be expected. So the top graph shows increasing doses of a drug with mono-exponential kinetics. The kind of equation that would apply is shown on the graph. And then he showed that the expected responses over time on the bottom are nonlinear. That at a high enough dose, there's a plateau, the Emax, and then the decline phase is, as Levy showed, nearly linear, but actually not quite linear because there's slight curvature to the data, and that can be captured with the Hill function. So these graphs are what we call signature profiles. What they are simulations of what's to be expected under the simplest of circumstances. So the simplest of drug actions is a simple direct effect. That's what succinylcholine does with muscle relaxation. That's what Wagner is showing in this graph. And that's what we see happening as an appreciation of a starting point, the simplest PK/PD behavior to be expected. Levy further identified the importance of and determinants of dose and elimination constant. So he provided a derivation that gave us an equation for duration of response. So look at the graph. It shows log of concentration versus time. So semi-log, it shows slope being K, and the C-min on the graph would be some minimal, the concentration at some minimal effect of the drug. So what you would expect to happen then is that bigger doses, the upper curve, would increase the duration of response. So he showed that that was proportional to log of dose. And then the graph is showing a K value. So you can imagine what would happen if a K value was smaller. That slope would be out flatter, and it would increase the duration. And that's reflected in the equation. 1 over K, if K is smaller, duration gets longer. So this is a fundamental concept of pharmacodynamics as well. For almost all types of pharmacodynamic effects, if you increase the dose, you increase the duration of effect. If you reduce the elimination rate, if you create a longer half-life, you expect a longer duration of effect. So that's reflected in these simple equations that Levy provided, but you see that this idea will be a cornerstone of all of pharmacodynamics. And it also should give you an appreciation of the importance of dose, the importance of the half-life of the drug in controlling pharmacodynamic effects. Well, there are more complicated types of pharmacologic responses. And Levy had these data for the effects of warfarin and showed more complex behavior that he called an indirect response. So you see on the graph plasma concentrations of warfarin, and you see on the graph the time course of inhibition of prothrombin complex activity. So it is an anticoagulant effect. So here what you see happening is that the maximum or minimum effect is happening at 2 or 3 days after you give the dose. Well, Levy identified the fact that what the drug is doing is blocking the synthesis of PCA activity in the liver. So the blockage is happening, and it takes time for that blockage to be expressed for the PCA in the blood. So that's a slowly acting type of behavior. So this idea of indirect responses, meaning the drug is acting either on a production process or a loss process, on one or the other side of a turnover process, led to our generalization that indirect response models apply to a wide array of types of situations. So this slide is showing 4 basic indirect response models. Model 1 is inhibition of K in. Model 2, inhibition of K out. Model 3, stimulation of K in. Model 4, stimulation of K out. In each of these cases, there are nonlinear differential equations that are needed to describe the models. In each of these cases, there's 3 components to the model. There's turnover. There's a K in and a K out for the basic turnover of the biological entity. There's pharmacokinetics. That's the C or CP function that actually represents any and all types of pharmacokinetics that could be involved. And then there's the Imax or Smax equation. That's basically the Hill function as inhibition or stimulation that governs the pharmacology of the drug. So we have PK pharmacology and physiology turnover as comprising these models. So these kinds of models will be used extensively when we get into more detail into pharmacodynamics. And when we get into more detail into pharmacodynamics, you'll be introduced to this categorization of 7 basic PD models that serve as a starting point for describing the simplest behaviors of a variety of drugs. So you'll see the application of these models to many types of drugs, be taught about the mechanisms of action, shown signature profiles, and be shown how to operate them to analyze data and provide simulations of drug action. So to summarize this lecture, PK/PD modeling is extensively used in assessing effects of small and large molecule therapeutic agents. PK, target binding, and turnover are key components of numerous models, and an array of basic and complex PK/PD models can be applied to diverse drugs. A lot more will be said on this topic later in the course. Thank you for your attention.